So thank you for sharing. Today we're going to continue to talk about carbohydrate metabolism. So last week, just kind of a summary to get us back all on the same page, we were looking at what happens with the structure of carbohydrates, how it gets transported in and around the body and into cells, and that first main pathway of carbohydrate metabolism, glycolysis. We also started talking about what metabolism is as a whole, and it's this balance between anabolism or anabolic processes. These are processes that take building blocks and take energy and make something new. Think of protein synthesis. We're going to talk about glycogen synthesis. We can talk about fatty acid synthesis, which Dr. Patterson will talk about next week. These are things that are going to be making bigger molecules. These are anabolic processes that require the building blocks, but also require energy. And that energy is derived from catabolic processes. Those energies, that process of breaking down a molecule, breaking down and releasing the energy stored in those chemical bonds, and storing them as ATP or as other energy sources for later use. So this balance between anabolic processes and catabolic processes is what makes up metabolism. Now, a cell can be in a catabolic state. This indicates that the net effect is going to be more catabolic processes occurring than anabolic processes. It's never all or none. There's always going to be some anabolism occur. You have some protein synthesis, some gene expression occurring, making those mRNA molecules that are going to require energy in those building blocks. You always have this happening, but when a cell is in a catabolic state, the net effect is going to be more catabolic processes breaking them down in order to release energy or provide resources for those essential tissues. When cells are in an anabolic state, that's when there's going to be lots of building blocks available, lots of energy available. Those cells are going to go into overdrive making new things, making cells, making proteins, making new molecules to help maintain homeostasis. In terms of carbohydrate metabolism, two hormones regulate this. Insulin is going to signal when there's lots of glucose available, carbohydrates available to be used for energy or as building blocks. Glucagon is going to signal when there's low amounts of glucose available, so cells are going to save it for those essential tissues, those red blood cells, those neurons. Epinephrine is also another one of these catabolic hormones, has a slightly different impact. It's a stress hormone, stress response, so it's going to release glucose into the blood and save it for those essential tissues for fight or flight, to allow those cells to survive and maintain homeostasis. But the net effect is the same. It's going to turn off those processes that use glucose in those non-essential tissues. So last week we talked about glycolysis. We looked to see that 6-carbon glucose molecule breaking down to those 2 3-carbon pyruvate molecules. Today we're going to see what happens before and after glycolysis. So before glycolysis, when we have excess glucose, we can store it as a glycogen synthesis, or when we're low on glucose and cells need to start breaking down that glycogen in order to maintain homeostasis in those cells. Then we're going to see what happens after glycolysis, looking at the fate of pyruvate. Pyruvate can be used in anaerobic processes, which is going to convert it to lactate, or it can enter the mitochondria and then be oxidized even further to generate more energy as part of in the TCA cycle and the electron transport chain. So we'll start to see what happens in the mitochondria today, and we're going to wrap up with that to look at the electron transport chain on Thursday. So, having said that, what I want you to focus on today for this lecture is to examine the role of thermodynamics. So I want you to understand those thermodynamic driving forces that allow glycogen synthesis to occur, and not listed here, but also the TCA cycle. I want you to examine, or be able to examine and speak to how hormone signaling is going to regulate glucose uptake, storage, and that regulation of glycogen, and the enzymes involved in how they're regulated by these hormones. I want you to understand why the mitochondria is so important, why we call it the powerhouse of the cell. I want you to be able to describe the structure and function of a key critical enzyme that links glycolysis and the TCA cycle, the pyruvate dehydrogenase complex. I want you to be able to review the TCA cycle, pull out some key important information for understanding how it generates energy, but also how it's regulated. And the regulation is going to always be an underlying theme when we talk about biochemistry and metabolism in this course. How are these enzymes turned on and turned off to regulate the flux or the flow of these molecules through these pathways? So keep these in mind as you go through this lecture and you start to study as well and prepare for the final exam. We use these learning objectives to help write the final exam, the questions. This is what we expect you to learn. So these are a key tool for you to understand what you should be studying as well. So let's start with a case study. Anyone here run or do any kind of endurance sports? A few hands throwing up. So if you've ever run, this might be familiar to you. But in this case study, talking about endurance running. So maybe imagine yourself preparing for an endurance running or endurance race. You're preparing for your first half marathon. Talking about your prep for your race with your running group, they start talking about carb loading. This is an unfamiliar term to you, so you start to look it up, and you see that carb loading is referring to increasing the consumption of carbohydrates in the days, maybe even a week, leading up to the race. Biochemically, I want you to think about what this is doing and how this can help someone perform for this endurance training or endurance event. So keep this in mind. We're going to revisit this case study later on in the lecture. But all this starts with glycogen. Glycogen is that key branched glucose polymer that we are able to store carbohydrates, or specifically glucose, within our cells. This is stored specifically within muscle cells and liver tissue. So this is the cells that are going to store glycogen for use, or store carbohydrates for later use. This is only going to store glucose when we have excess glucose. So think about when we're going to have excess glucose. When you've consumed a meal, and you have lots of food, lots of carbohydrates coming in, there's lots of glucose available. Or when you have lots of energy available and you don't need it anymore, that regulatory mechanism is going to turn glycolysis off. Remember when pyruvate increases, pyruvate and ATP levels will turn off pyruvate kinase, start to back up the glycolytic pathway. Phosphofructokinase is going to be inhibited as well as those molecules build up. And then we're left with hexokinase. Hexokinase catalyzes that first step that converts glucose into glucose 6-phosphate. In most tissues, this is going to trap it within the cell. This is that key regulatory point deciding the fate or what happens to glucose in that cell. If hexokinase is turned on, and converting glucose to glucose 6-phosphate, it has to be used by that cell. If glycolysis is turned off, maybe you can use another pathway, like the pentose phosphate pathway, to make those riboses that are useful for creating DNA, those nucleotides. Maybe it's used in some other pathways. However, as glucose 6-phosphate builds up, remember, this also inhibits hexokinase. So in most tissues, that inhibition of hexokinase is going to allow glucose to move back and forth in and out of the cell through those glucose transporters, through facilitated diffusion, across this concentration gradient from high to low. If the concentration of glucose in the cell is higher than it is in the blood, it'll move back across the membrane, but only if it's as glucose. If it's been phosphorylated, it can't move through that glucose transporter. So in tissues where hexokinase is active and it's converting into glucose 6-phosphate, that glucose 6-phosphate needs to be used. In particular in the liver and muscle tissue, this glucose 6-phosphate can be used to make glycogen. And this is how we store glucose as that branch carbohydrate. The first step of this pathway is to isomerize glucose 6-phosphate into glucose 1-phosphate. So this is carried out by the enzyme phosphoglucomutase. This process, whenever you see mutase within an enzyme, remember A's refers to an enzyme. A mutase is often a type of enzyme that carries out an isomerization reaction. It converts that molecule to essentially an isomer of itself. Glucose 1-phosphate and glucose 6-phosphate are isomers. They have the exact same molecular formula, except their functional groups are now in a different order. They're on different sites. So when we have cells that have insulin signaling is indicating that glucose uptake, so let's maybe think about muscle cell in particular, that glucose is going to be phosphorylated. It can be used in glycolysis. However, if glycolysis is turned off, it has to be used in another reaction now. So this is where now a new enzyme, phosphoglucomutase, can react with that glucose 6-phosphate and convert it into glucose 1-phosphate. Now, this isn't a simple reaction of just moving the phosphate from one group to another. This actually has a much more intricate pathway, and we're going to kind of highlight just some of the main features of that pathway. In that active site, there's a phosphorylated serine that's able to be part of the enzymatic reaction. This is a critical part of that enzymatic reaction. When glucose 6-phosphate is in that active site, that hydroxyl group on the anomeric carbon on C1 can attack that phosphate, breaking that bond, generating a bisphosphate molecule, glucose 1,6-bisphosphate. Bisphosphate meaning that it has two phosphate groups on it now. This is an intermediate of the reaction. Remember, for an enzymatic reaction, for that to occur, the enzyme needs to be regenerated. So in this reaction, that serine now, that's not phosphorylated, can attack the other phosphate group at position 6, breaking that bond, and regenerating the enzyme, so phosphorylated serine in the active site. But the result is that glucose now has a phosphate group on its position 1 instead of position 6. That phosphate group has moved, even though it's not the same phosphate, but we generated an isomer of that molecule. And now glucose 1-phosphate can enter into the glycogen synthesis pathway to produce and store glycogen. Now this is an anabolic reaction. We are making a larger molecule using building blocks of glucose, and then it requires energy. Now, the energy in this pathway is coming not from ATP, which we might more commonly see, but from a different trinucleotide, phosphate trinucleotide, uracil triphosphate. All of the nucleotides can be used for energy interchangeably. It's always going to depend on the preference of that enzyme and what's going to fit within its active site. ATP is a common one we might see in many reactions, but GTP, UTP, CTP, TTP, all of these can be used in enzymatic reactions depending on the enzyme. In this case, UDP glucose pyrophosphorylase is going to catalyze the conversion or the transfer of a phosphate group from transfer of actually UDP to glucose to activate it. But glucose 1-phosphate also had a phosphate group. why can't it just use glucose 1-phosphate? That phosphate bond does have energy in it. Why can't we just separate the phosphate group and move it onto glucose? Why do you think this might be a challenge? The Menti is up now. Why can't this enzyme just use glucose 1-phosphate? So starting to see some responses coming in. I'm seeing it won't fit into the active site, not enough energy for the GP bond compared to trinucleotide, yes. Here's a great response, I'm going to throw up on here. May have to do with the energy coupling and the activation energy required for the reaction to occur. So we're thinking about the energy in the reaction. Thinking about that reaction and that energy in that phosphate bond is not really enough to carry it over, to activate it and make this what we call a favorable reaction, to have a negative change in the Gibbs free energy. That hydrolysis ends up being a reversible reaction with a delta G of equal to about 0 in the cell. It's really hard to make a molecule when you have a reversible reaction where it's going to be made and then unmade in the same response. So in order to overcome this, we need to couple it with a different reaction. So thermodynamically, this has to be coupled with other enzymatic reactions. So this is where UTP comes in. We are adding energy to this reaction to make it a more favorable reaction and move in that forward direction. Glucose 1-phosphate can now attack that alpha phosphate on UTP, breaking the bond and generating UDP glucose, and importantly, a pyrophosphate. This is a common strategy you're going to see among metabolic pathways and enzymatic reactions, the generation of a pyrophosphate, 2 phosphate molecules. So this is a double, you're breaking essentially 2 phosphate bonds. That pyrophosphate itself then gets hydrolyzed to 2 inorganic phosphate molecules, releasing even more energy. So this couples the reaction together and makes it favorable. Can you have the reverse reaction occurring if pyrophosphate is never there? If it gets used up immediately, hydrolyzed and broken apart, is there any chance for this reverse reaction to occur? The reverse reaction requires pyrophosphate as a substrate. If there's no pyrophosphate, no reverse reaction can occur, so it drives the reaction forward. It pulls it in that forward direction, allowing us to synthesize glycogen. And this is what happens with pyrophosphate. It can be stable, it can hydrolyze on its own, but in the cells, we have an enzyme that breaks apart pyrophosphate almost immediately. So it is gone. And when it disappears, it pulls that reaction forward. So the final step, now that we have UDP glucose, is that we can add that UDP glucose onto the glycogen chain. Remember, from the non-reducing end. This is going to catalyze the formation of a new alpha-1,4 glycosidic bond, building that glycogen molecule piece by piece by building those building blocks. This reaction is just going to be a very similar reaction again. You're going to have that hydroxyl group attacking that anomeric carbon from the alpha position, forming that new alpha-1,4 glycosidic bond and releasing UDP. So we've taken what was essentially a molecule that has 4 phosphates. We had 1 phosphate and glucose 1-phosphate, 3 phosphates and UTP. We removed 2 of the phosphates and hydrolyzed them to generate enough energy to drive the reaction forward. And now we're left with UDP at the end as well, the other 2 phosphates. Just as a bit of a review, labeling these carbons, and labeling the carbons within the glucose molecule. Here's your oxygen. Look for the carbon next to it that has the hydroxyl group. That's the anomeric carbon. So here's C1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So this is a 1,4 bond. Because it's under the ring, it's an alpha-1,4 linkage. Coupling all these reactions together makes this a very energetically favorable reaction. It's going to release energy to help drive that reaction forward. And remember, whenever we have energetically favorable reactions that are essentially irreversible, those are the ones that get regulated. So we're going to see how that regulation occurs in a few slides. But before that, we're going to talk about the reverse process. How do we break this apart? Because this is an irreversible reaction, we need a separate enzymatic reaction to now break these bonds and break it apart, because these reactions are not reversible. We call that process glycogen breakdown or glycogenolysis. This is a phosphorylasis reaction where that free phosphate can come in and release those glucose molecules one at a time from those ends of those branches. That reaction is taking that free inorganic phosphate, allowing it to react at the anomeric carbon, breaking that bond, that glycosidic bond, and releasing now glucose 1-phosphate, and leaving the remaining branch of our glycogen molecule. So this breaks apart those glucose molecules one at a time, but what we're left with is glucose 1-phosphate. Now this reaction is reversible, that converting glucose 1-phosphate to glucose 6-phosphate. That phosphoglucomutase, depending on which substrate is going to be, or which either its substrate or product is going to be in higher concentration, is going to depend on which way the reaction moves. If we have lots of glucose 1-phosphate, they'll start converting it to glucose 6-phosphate, a reversible reaction. Now in the muscle cells, assuming that we need glucose to fuel energy or fuel activity within our muscle cells, those tissues are going to start to use, those cells are going to start to use it for energy and breaking it down in glycolysis or maybe other pathways. However, in our liver cells, we have a unique enzyme, glucose 6-phosphatase, that's able to remove the phosphate molecule, generating glucose, and now glucose can move across its concentration gradient from high to low back into the blood to maintain blood glucose levels. Sorry, there's a response. But here's a question for you. Glycogen is stored in the liver and muscle tissue. There's probably about 300 to 500 grams of glycogen in muscle tissue, depending on muscle mass, while the liver can store about 80 to 100 grams of glycogen. Which of the following is most correct? Which of the following is most correct regarding the use of these carbohydrates stores to maintain homeostasis? Let me just get the Menti up. Is it A, during fasting, muscle glycogen will be broken down to fuel neurons? B, liver glycogen is primarily used to maintain blood glucose levels? C, both liver and muscle glycogen are equally important in maintaining blood glucose levels during prolonged fasting? Or D, muscle glycogen is the main source of blood glucose during endurance exercise because it's of its large storage capacity? What do you believe to be the most correct answer? Survey says majority going with B. Liver glycogen is primarily used to maintain blood glucose levels. There's also a proportion going with C or D, thinking about the uses maintaining blood glucose, the importance of muscle glycogen potentially maintaining blood glucose levels. The correct response here is B, and it all comes down to that ability of the liver to release glucose back into the bloodstream. That enzyme, that glucose 6-phosphatase, is a unique enzyme within the liver. Only liver cells express this enzyme, so only liver cells can convert glucose 6-phosphate back to glucose, allowing it to now enter into the bloodstream. This makes the liver critical in maintaining blood glucose levels because of this gene expression or this protein expression. In the muscle cells, even though glycogen is present within the muscle cells, it gets broken down to glucose 6-phosphate, and it can only be used by that muscle tissue. The muscle tissue must use it for glycolysis or for other metabolic pathways, but primarily is going to enter into glycolysis to fuel activity or movement. So this is the key thing in trying to balance these two processes or two uses of glucose. Depending on the tissue type, they are going to respond to hormones slightly differently. When someone is in an anabolic state or when insulin is expressed, there's lots of glucose. That glucose is going to be used either to fuel catabolic processes or storing the excess as glycogen. However, when blood glucose levels are low, glycogen is going to start to break down, or glucagon is going to signal the breakdown of glucose, allowing it to maintain blood glucose levels for those essential tissues. Those tissues are the red blood cells, because they don't have mitochondria. They're going to rely exclusively on glycolysis. And neurons that need glucose to cross that blood-brain barrier, and are going to rely on glucose as a fuel source as well. Epinephrine is also going to be one of these, what we call a counter-regulatory hormone, or counters the impact of insulin. And this is more about kind of a stress response, the fight or flight response. During times of stress, all of those stores are going to start to break down to increase blood glucose levels. This is to fuel essential processes to respond to whatever stress is occurring. So we see the stress response as being critical in regulating blood glucose levels, and it regulates them by regulating glycogen. So glycogen, again, found in the muscle and the liver cells, and the balance of this degradation is going to be regulated by the balance of these two hormones. When we have insulin up, lots of blood glucose levels, there's going to be a net production of glycogen, storing all that excess glucose for future use. When we have low blood glucose levels or responding to stress, that glycogen is going to get broken down, adding glucose to the bloodstream in order to respond to the stress or to maintain blood glucose levels. So this regulation is really key to maintaining the levels of glycogen in all these tissues. I have this signaling pathway here on the right. Notice the information-only icon. You're not expected to memorize this, but I do want you to understand how these signaling cascades regulate these pathways. And you're going to see some familiar players that have come up before. Focusing on insulin, insulin is going to regulate glycogen synthesis through two main ways. It's going to regulate those two key enzymes, glycogen phosphorylase and glycogen synthase. So the one enzyme that's going to break down glycogen and the other enzyme that's going to act to build up glycogen. It does this through a signaling pathway where insulin binds to that insulin receptor. Okay, stop moving. Insulin binds to the insulin receptor, and it initiates a signaling cascade that results in the activation of PP1, or this is a protein phosphatase. That protein phosphatase is going to dephosphorylate glycogen synthase, and it's going to put it into its active state. When glycogen synthase is phosphorylated, it's going to be inactive. So insulin regulates the enzyme that takes it off, activating glycogen synthase, allowing those cells now to use the glucose that's available to make glycogen. On the other side, it's going to dephosphorylate glycogen phosphorylase. So that enzyme that breaks down glycogen is going to turn it off. So by regulating these two processes, turning off glycogen breakdown, turning on glycogen synthesis, is shifting the balance to building up glycogen. Epinephrine and glucagon have an opposite signaling cascade. They bind to those GPCRs that we talked about, and they're going to signal the activation of a kinase, protein kinase A primarily, that eventually leads to the activation or the phosphorylation of glycogen phosphorylase. This phosphorylation now activates this enzyme and turns it on, allowing it to start to break down glycogen and releasing glucose. So we see the balance of these two pathways activating or inhibiting these key enzymes, turning on or turning off these processes. Other regulatory mechanisms include allosteric regulation. So remember, the products of a reaction often inhibit the enzyme itself. So glucose and glucose 6-phosphate and ATP are going to, glucose 6-phosphate and ATP are going to inhibit this process. So when you have lots of energy or lots of glucose 6-phosphate, you're going to inhibit glycogen phosphorylase, signaling the synthesis of glycogen. And conversely, if you have a low energy state, lots of AMP around, so that triphosphate's now gone to a diphosphate, now gone to a monophosphate, it's going to signal glucose, it's going to turn on this process to start to release energy. So it's a balance between these two hormones and maintaining this anabolic and catabolic states or processes. And as I mentioned, that muscle and liver glycogen are used for different purposes. In the liver, that glycogen is going to get broken down, released or converted into glucose, and then glucose can cross this, the cell membrane and enter in the bloodstream across this concentration gradient. In the muscle, that glycogen and that glucose can never leave once it's in that cell, and it's only going to be used for energy within the muscle tissue. So, I got a question for you then. If muscle cells contain glycogen, a form of carbohydrates in animal cells, why does the meat we consume that comes from muscle tissue in animals, why does it not contain any carbohydrates? We always think of it as a protein source, fat source, never as a carbohydrate source. But we just talked about how muscle cells have carbohydrates. Why do you think that is? Is it that glycogen is used up by the muscle cells, resulting in the loss of the carbohydrates in meat? The carbohydrates in muscle cells are converted into fats during the process of rigor mortis, which results in no carbohydrates present in the meat we consume. The carbohydrates in muscle cells are not digestible by humans and are therefore not present in meat, or they're just fiber. The carbohydrates in muscle cells are present in meat, but are in such small quantities that they're not significant and they're never going to make it on the nutritional label. Essentially, we don't care about it because it's such a small amount. What do you think? Ooh, a little bit of indecision within the group. Seeing maybe it looks like a slight majority going with A, that glucose is used up by the muscle cells. Next best response is that the carbohydrates are present in meat in such small quantities that they're not significant enough to be detected. And then a good chunk of people also thinking that they're converted to fats during the process of rigor mortis. Now the correct answer is A. Let's talk through this. Think about what's happening within an animal when it's slaughtered. That is death. That's going to stop the heart, stop blood flow. In the blood is where oxygen is going to be transported. So you're stopping oxygen transport to those tissues. And we're going to see how important oxygen is for mitochondria and generating energy in that way. But you're also going to stop glucose flowing and fuels flowing through the cells. Those cells do not die instantly. Those cells will use all of their tissue, all of their resources available to survive for as long as possible. It's only when they exhaust all their fuel sources that they will begin to undergo cell death, or in the process of converting from a muscle cell into what we would call meat. So that process means that in those muscle tissues that now are starved of oxygen, starved of glucose and fuel coming from the blood, they're going to start to use their internal resources. Glucose levels are going to drop, and that's going to signal the breakdown of that glycogen. As that glycogen is broken down, it's used in glycolysis within those cells, so it's going to get used up to maintain homeostasis for as long as possible. Only when that glycogen has been broken down completely and exhausted does that tissue now undergo essentially cell death and that conversion into meat that we then consume. So this process is critical. It also results in the buildup of lactate and the acidification of our meat, of the meat that's part of that process that occurs. So this process means that the muscle cells we consume no longer have any of those carbohydrates present because those cells have used them all up. But I said there's another tissue that has carbohydrates, livers. If you consume liver meat, and you look at the nutritional label, you will see carbohydrates listed on that. This is indicative of how much carbohydrates are stored within the liver, allowing us to maintain our blood glucose levels. That even during that process of cell death, there is still some leftover carbohydrates that have occurred through that acidification process. The liver cells do not exhaust all their carbohydrates. So if you are a carnivore and you're looking for a source of carbohydrates, liver is probably a good source as well. What questions do you have with that before we move on? Raise your hand if you have a question. We'll come around. Yeah? You want to bring the mic or no? So the question is a great question. How do other carnivores then start to store carbohydrates? If they're only consuming meat, where is their source of carbohydrates? So we're going to talk about that in a couple weeks, where we talk about other processes to store carbohydrates and store in gluconeogenesis. So Dr. Patterson is going to talk about all the other ways in which we can generate our own glucose within our cells. Great question. Yep? So the question is, and yes. The question is then, I'm going to go with an animal cell, an animal that's consuming something that is still alive. Yes, there's potentially to be in that muscle tissue that's from an animal that has not yet perished. Yes, there is potential for there to be carbohydrates within those muscle tissues. Yes. All right. Great questions. And I want you to start to think about how we think about metabolism and we think about biochemistry in the context of your everyday lives. This is part of the reason why I keep bringing up these case studies, because I want you to see how this applies and how you can use this information to help answer these questions or critically evaluate or appraise things that you're going to encounter throughout your life. Biochemistry is fundamental to how we understand everything. And this is why this ends up being so useful for us. So we looked at glycogen and what's happening to all that extra glucose, or how we start to break down glycogen to fuel glycolysis. Now we're going to look at the other end and look at what happens at pyruvate. So remember, when that glucose goes through glycolysis, that 6-carbon glucose molecule gets broken down to 2 3-carbon pyruvate molecules, releasing some energy, releasing some NADH. Now that pyruvate has multiple fates depending on what type of cell and what's available around it. In cells that don't have a mitochondria, or when there's no oxygen present, that pyruvate is going to undergo a process called anaerobic metabolism, where it gets converted into lactate. That process takes pyruvate, using an NADH, generating a lactate molecule. So this is the conjugate base of lactic acid, but generating lactate. This is a key step allowing glycolysis to occur. In a cell that does not have a use for these NADH molecules, those cells are going to start to exhaust their supply of NAD+. So even if there's lots of glucose, if there's no NAD+, glycolysis can't keep going. So in order to allow that to keep going, this side reaction now converts that pyruvate to lactate, regenerating NAD+, to allow glycolysis to keep going. So in red blood cells, where there's no mitochondria, that pyruvate needs to be converted into something. You also have to think that if pyruvate continues to build up, it's going to turn off pyruvate kinase and turn off the glycolytic pathway. That pyruvate needs to go somewhere in order to allow glycolysis to occur. So this process not only uses up the pyruvate, but it also regenerates NAD+ for glycolysis to keep going. So red blood cells that don't have a mitochondria rely exclusively on this process in order to maintain cellular homeostasis. When you think about other tissues that may be deprived of oxygen, so someone who may be experiencing a stroke or a heart attack, those tissues will rely on this process as well in order to maintain homeostasis as long as glucose is available for that short period of time. That's why lactate in the blood becomes an indicator of potential hypoxia or hypoxic states within those tissues or where cells that might be deprived of oxygen. When pyruvate is available, when mitochondria is available and oxygen is available, that pyruvate is going to be able to enter into the mitochondria, and we're going to see what happens to it as part of the TCA cycle just after the break. I do want to go on a tangent. How many people have heard of lactic acidosis or lactic buildup when you're exercising? We commonly refer to this as kind of lactic acid buildup or lactic acidosis, but I do want to caution and say that lactate is never in its acid form. When we convert pyruvate to lactate, it's always deprotonated, and because that acid group has a pKa of 3.8, our tissues or cells are usually around a pH of 7, 7.35 in the blood, it is never going to be protonated and never result in acidosis. It cannot donate a proton to the solution to cause acidosis. When you're seeing some sort of a metabolic acidosis, like when you're exercising to exhaustion or to fatigue, that acidosis is the result of the breakdown of ATP. When ATP is hydrolyzed, so all those tissues using ATP to help your muscles flex, to help you lift, to help you run, that buildup or breakdown of ATP generates free protons. Those protons result in a change in the pH of your blood, of your cells, of your tissues, resulting in acidosis. So if the balance of ATP breakdown is faster than the ATP genesis or synthesis that we're going to talk about a little bit later, you're going to start to see this acidosis developing. What we talk about when we see lactic acidosis, we're seeing an active acidosis occurring. We're also seeing lactate present. So that's an indicator of what might be happening within the cell, but it's not the cause of that acidosis. Now that lactate also needs to go somewhere. If lactate builds up, lactate dehydrogenase is going to turn off as well through product inhibition. So that lactate in those red blood cells, in those tissues that are using anaerobic glycolysis, anaerobic metabolism, needs to move through that process. So it's going to enter into the blood, move to the liver, where it's going to get converted back to glucose as part of gluconeogenesis. And Dr. Patterson's going to talk more about this in the next couple of weeks, so I'm going to leave this for her discussion, but just know that that lactate then has to be used somewhere else as well. So coming back to our case study, you've recently become, if you remember from the beginning of our lecture, about 45 minutes ago, you've recently begun endurance training, running, and you're preparing for your first marathon. You're kind of chatting with your running group, and everyone's talking about carb loading. You're unfamiliar with this term, but you look at it and you see that it refers to increasing carbohydrate concentration or consumption in the days leading up to a race. So biochemically, what do you think this is? What do you think this is doing, and how can it help your performance? How could this help you if you're participating in some sort of endurance training? So lots of great responses coming in, seeing the more carbohydrates, the more can be used as energy to help in endurance, yes. By building up glycogen stores in the muscle, absolutely. A lot of glycogen will be stored and then it'll be broken down during the race to give you energy. Absolutely. This is all about increasing your glycogen stores. When you're increasing carbohydrate consumption, the concentration of blood glucose is going to increase during those fed states, and it's going to be stored as glycogen. If you're doing it over the few days, you can increase your glycogen stores by maybe 15, 20% more than what you'd normally expect. During a race or during endurance exercise, you can start to use this glycogen now in your muscles to fuel your metabolic processes. Dr. Patterson's going to talk more about kind of what those different processes and why that's also important. So glucose is a really important fuel source. But just briefly, the four main fuel sources that you're going to see within cells are going to be ATP. You don't have a lot of ATP in your cells at any given time. You generate it as you need it. So this is only going to last probably your stored ATP or your resting concentration of ATP will probably only last a couple seconds in each cell during exercise. The other store that we've talked about is creatine phosphate. So you can store some of that extra ATP as creatine phosphate, and use that for energy. And that gives you probably an extra few seconds of energy during exercise. The other two main sources are going to be your carbohydrates and your fats. Your fats are one of your biggest fuel sources. Think of it as your gas tank. You're going to talk about those two different processes that kind of break down energy. We're going to focus on carbohydrates now, but you're going to see later on with fats as well. Fats can generate a lot of energy, but it's a slow process. It doesn't generate it very quickly. Glycolysis and carbohydrate metabolism can generate a little bit of energy, relatively less, but it generates it very fast. This makes it a very important fuel source when you're doing any kind of intense or endurance energy or endurance exercise. When you're walking around right now, my fuel that I'm using to fuel my processes, my muscles that are allowing me to speak, my muscles that are allowing me to move, these are mostly going to be fatty acids. If I were to start exercising, in order to make up the difference to generate enough ATP, I need to start using those carbohydrates. We'll talk more about this in the next few weeks, but just know that carbohydrates become an essential fuel source to fuel exercise and fuel activity. So with that, we'll take a break. We'll come back at 10:07, and yeah, we'll wrap up. We'll see what happens to the pyruvate once it gets into the mitochondria. Back to this question here. Glycogen is stored in the liver and muscle tissue. There's 300 to 500 grams of glycogen in muscle tissue, depending on muscle mass, while the liver can store 80 to 100 grams of glycogen. Questions about why there's so much more glycogen in muscle, but when you consume food later on, like liver, if you consume liver meat, it still has glycogen left, but muscle is exhausted. This comes down to the relative size of these two tissues. Your liver compared to all the muscle mass in your body. You have much more muscle than you have liver. So on a per cell basis, the liver has more glycogen per cell, per hepatocyte, whereas muscle cells, there's so many muscle cells, so each individual muscle cell has less glycogen in it, but there's just so much more muscle within your body. So that's why there's more glycogen stored in muscle, even though each cell has less. Now coming back to this question here, if muscle cells contain glycogen, why do they exhaust it, and why do livers not exhaust their stores? Liver tissues are designed to release and maintain blood glucose levels. So when glycogen is getting broken down, when during that stress response or during when that cell is dying, that glycogen is being primed to be released into the blood, but blood flow has stopped. So thinking about that transport across those glucose transporters, the concentration in the liver cell is getting very high. It's starting to transport it into the blood, but blood flow is not moving anymore. So the local concentration of glucose around the liver is going to be relatively high, keeping the glucose within the liver and then stopping glycogen breakdown because glucose concentration is increasing. The liver cells do not use glucose for energy, or will not use glucose for energy unless absolutely necessary, so they will actually maintain some of that glucose when the cell gets converted into meat. So those two things to just clarify those 2 points. Now continuing on, let's look at what's happening to what happens to pyruvate. We've seen how pyruvate can be broken down to lactate through anaerobic metabolism if a mitochondria is not present or if there's no oxygen. But what happens in cells that do have a mitochondria? That pyruvate can now move from the cytoplasm, where the glycolysis is occurring, move into the mitochondria, where it can be broken down or oxidized within a TCA cycle, and then generating energy in the electron transport chain. This is in part due to the structure and the nature of the mitochondria. That mitochondria is a double membraned organelle. That double membrane has a permeable outer mitochondrial membrane that's going to allow molecules to move in and out of that and through that membrane. There's lots of pores that allow molecules to move through that membrane back and forth between the cytoplasm and that will become that intramembranous space. Is that inner mitochondrial membrane that is very impermeable. Things cannot move through that membrane very easily. This allows it to generate some concentration gradients, and we see this as being important for the electron transport chain. The lumen, or the matrix of the mitochondria, that space in the middle, is where all of these enzymes are going to be present. All the enzymes involved in fuel metabolism, the TCA cycle enzymes, enzymes involved in the breakdown of fatty acids that Dr. Patterson will talk about next week. The enzymes for the electron transport chain are going to be along this inner membrane. And then ATP synthase as well. So this is the process where all of that oxidation and fuel metabolism can occur. Everything is concentrated within this one organelle to allow these processes to occur. But in order for this to occur, pyruvate needs to get in there. And pyruvate does that through, one, moving through those porons to get into that intermembranous space and into that mitochondria. And then it needs to be converted into something that's useful. This first step is the pyruvate dehydrogenase complex. This is a critical step because it connects glycolysis to the TCA cycle. If this step is not working, if if this enzyme is turned off, these two pathways are disconnected and separated. So this is a critical step that connects these two processes. Looking at the structure, we're going to see dehydrogenases a lot. We've seen a couple already, lactate dehydrogenase. Here's pyruvate dehydrogenase. Whenever you see a dehydrogenase, you should be immediately thinking that some sort of NADH is going to be involved. So this is a very large complex. It has three enzymes connected in a complex. I'm going to circle them here. Maybe hard to see. And then we see multiples of these enzymes coming together to form this large complex. This complex has a lot of different steps in it and a lot of cofactors. And some of these cofactors are going to start to become more familiar to you. Thiamine pyrophosphate, TPP. This is also vitamin B1. Lipoamide. FAD, which is vitamin B2. Coenzyme A, which is vitamin B5, and NADH, which is vitamin B3. Almost all your B vitamins that you're going to consume have required roles within energy metabolism. This is why they're important as cofactors and why you need to consume them within your diet or potentially through a supplement. Looking at these cofactors, TPP, lipoamide, and FAD are prosthetic groups. So remembering from the first half of the course, a prosthetic group is going to be a cofactor that's permanently or covalently attached to that enzyme. It's not going to be released. And then coenzyme A and NADH are coenzymes. These are cofactors that can be released. So they're going to be interacting with the enzyme, participating in the reaction, and then they leave and a new coenzyme can come in for the next reaction. So we see these different cofactors that are going to be critical for allowing this process to occur. Now, what's an advantage of having all these subunits forming a complex together? Why is this advantageous? Why does a cell evolve this process? Thinking about proteins and how these processes evolved, this is an evolutionary pathway where in most cases, there's something advantageous about the way this protein complex is formed or this process is formed. What would be advantageous for making an enzyme subunits forming a large complex or different enzymes forming this complex together? Seeing the process coming in, there's a couple great responses coming in. Localizes everything and allows it to occur faster. Let's see, what else? Come here. What else do we see here? Increase more efficiency, regulation, easier since they're together, absolutely. So all these things kind of make it more efficient. Thinking of, as a cell evolved, having these processes occurring in close proximity to each other, either localizing these enzymes within a single organelle or bringing them together in a protein complex, makes these processes occur much more efficiently and faster. And that could be evolutionarily advantageous to a cell, because thinking of a cell trying to survive, if it can generate energy faster or potentially save energy by regulating this process easier, that gives it an advantage in growth and survival within a very complex environment. So looking at this reaction, this is actually 3 enzymatic reactions in one. Overall, this is going to convert pyruvate into acetyl-CoA, and it's a decarboxylation reaction. We're taking this 3-carbon pyruvate, which was the product of glycolysis, and we're converting it to a 2-carbon acetyl-CoA molecule. That 1 carbon, this is the first oxidation where we lose carbon dioxide. We're releasing carbon dioxide in this process. Because of this complex reaction that's occurring, all those cofactors are going to participate. But overall, this is a very energetically favorable reaction. It's going to release about 33 kilojoules of energy. A lot of the other reactions we've seen that have been energetically favorable have involved some sort of a triphosphate, ATP, UTP. This one doesn't. Of the cofactors, coenzymes, substrates, we don't see any energy involved. What contributes to this potential, the favorability of this reaction? I don't have a Menti for this, just ideas. Anyone want to shout out? What do you think? What's making this a very favorable reaction? What happens to carbon dioxide? Carbon dioxide here is the key. Is carbon dioxide typically a gas or a liquid? Gas, right? It can be dissolved in liquid, but it gets dissolved as bicarbonate. So it's either going to be gaseous, disappear immediately, or it has to be converted to bicarbonate and leave, because it's not soluble on its own. This means that this product of the reaction is gone immediately. It disappears. And when a product of the reaction disappears, it drives the reaction forward. We saw this with pyrophosphate as well. That pyrophosphate molecule, because it gets hydrolyzed immediately, it pulls the reaction forward and it makes it an irreversible reaction. Likewise here, carbon dioxide, or the release of carbon dioxide, pulls this reaction forward because it disappears immediately. It gets used up. We see this as the potential driving force thermodynamically in many reactions as well within metabolism. This carbon dioxide release, or pyrophosphate release, that's going to get used up immediately, a relatively unstable product of the reaction that disappears. It means that the reaction essentially becomes irreversible. So it's a common theme you're going to see. You'll see it here, and you're going to see it again in Biochem 311 if you take it with me in the winter or next year. Looking at this reaction, three enzymes. Three enzymes coupled together occurring through a 5-step reaction. One enzyme is going to catalyze the reaction. A second step is going to regenerate it. A second step is going to regenerate that. Another step is going to regenerate that, resulting in the final reaction that generates all the enzymes. Focusing here, what I want you to know is the overall flow and the three enzymes and what they're working, how they're working, but you don't need to memorize this reaction mechanism. Starting with pyruvate, E1, or the first enzyme in this complex, catalyzes a decarboxylation of pyruvate, releasing carbon dioxide, so driving this reaction forward and transferring those two carbons to its cofactor. Those two carbons now are going to move through a redox cycle in enzyme 2, where they're going to be passed to an oxidized lipolysine, and eventually those two carbons are going to be transferred to coenzyme A, forming acetyl-CoA. Acetyl-CoA can leave, and now you have your two main products of the reaction, carbon dioxide, acetyl-CoA. Now, what you're left with is a reduced coenzyme that needs to get back into this oxidized form. So we have a couple redox reactions here that help generate this. In enzyme 3, we have that FAD molecule that's going to participate in that redox cycle, oxidase lipolysine, generating FADH2. And now in order to regenerate the E3 enzyme, you're going to transfer those electrons to NADH, regenerating FAD. So overall, this reaction is going to regenerate this molecule through these 5 different steps. The enzyme is ready to catalyze another round of the reaction, and we are left with the 3 main products of the reaction: CO2, acetyl-CoA, and NADH. NADH is going to come up again and again within the TCA cycle and in the electron transport chain. Think of NADH as a surrogate for energy. We're going to talk about on Thursday what happens to NADH in the electron transport chain, but this is free energy within the cell. It just needs to convert it to ATP as needed. Acetyl-CoA is a really important molecule as well. So it's another way to think about free energy. Acetyl-CoA, or acetyl coenzyme A, is an acetyl group, a 2-carbon group, that 2-carbon group that came from pyruvate, bound to this coenzyme A. Some of the structure might be familiar. You should see nucleotide, so the ribose, nucleotide, diphosphate with some other phosphate molecules is here. This is a complex vitamin that you consume in your diet that you need in order to generate acetyl-CoA. Often, though, when we write it out, you're going to see it just listed as CoA-S, just to simplify the structure. So whenever you see acetyl-CoA, you see this CoA with this acetyl group, just know there's a much larger molecule behind it. This molecule, the hydrolysis of acetyl-CoA, removing it, generates a lot of free energy. That free energy is what is going to be used within the TCA cycle to catalyze this reaction that we can extract to generate or transfer that energy to those NADH molecules. So this process is critical for linking these two pathways together, glycolysis and the TCA cycle. That pyruvate dehydrogenase complex is a complex molecule of 3 enzymes catalyzing a 5-step reaction. It's going to convert pyruvate that's coming from the cytoplasm, allowing it to move into, once it's moved into the matrix, catalyzing that conversion into acetyl-CoA. We can call this overall a redox decarboxylation. That decarboxylation, or the release of CO2, is what's going to drive the energy, drive the reaction forward, and that energy is going to be captured in two places. One, within acetyl-CoA, and two, within NADH. And both these are going to release energy, or provide energy that can be used to fuel the generation of ATP later on. But as we mentioned, this is a very large negative delta G. All the reactions that are going to be very favorable, essentially irreversible, those are the ones that tend to be regulated. And pyruvate dehydrogenase complex is like that, is heavily regulated as it connects these two pathways and is energetically favorable. In general, we can start to see also the products of all these reactions start to regulate their pathways and turn them on and turn them off. We've seen pyruvate turn off glycolysis. We're going to see now acetyl-CoA and NADH. It's going to turn off PDC, pyruvate dehydrogenase complex. ATP is going to turn off everything. As these levels of these molecules, the buildup of these products in the reactions build up, it starts to turn off molecules or enzymes within the pathway to start to turn it off in order to allow these to be used for different things. When you turn off the TCA cycle, this can be used for fats. You'll see that next week. When you turn off PDC and glycolysis, glucose can be stored as glycogen. So turning off this pathway allows these molecules now to be stored for energy later, saving them for when there's a time of need. So this feedback control, or the control of the PDC, is going to occur through multiple mechanisms, but they should start to be familiar. We're going to see product inhibition, so acetyl-CoA and NADH. They're going to bind to and inhibit their enzymes, the relative enzymes, as a product. So acetyl-CoA is going to inhibit E2, so this E2 enzyme, by either binding to the active site or through some allosteric regulation. NADH, which is a product of the E3 reaction, is going to bind to and inhibit that enzyme. This will turn off the process and turn off the enzyme, causing it to back up, and then pyruvate is going to start to build up and start to turn off glycolysis. We also start to see post-translational modifications. So those signaling cascades, those kinases and phosphorylases that turn enzymes on and off. Pyruvate dehydrogenase complex, and in particular E1, is going to be a site of post-translational modifications. When it's phosphorylated, it's going to be inactive or turned off. When it's dephosphorylated, it's going to be active and turned on. Tellingly, ATP is going to be a regulator or a substrate in this process. So I want you to think about this. I'm going to put this down to you. Use some deductive reasoning. Predict the effect, either stimulating or inhibiting these processes, these enzymes. So what is going to happen when there's lots of acetyl-CoA, NADH, pyruvate, ADP, or calcium within the cell? What impact is it going to have on that pyruvate dehydrogenase complex? And then likewise, what's happening to PD kinase and PD phosphatase? So take a second and think through that. What is going to happen when there's lots of acetyl-CoA, NADH, pyruvate, ATP, or calcium within the cell? What's the impact on the pyruvate dehydrogenase complex? Think about what this means for the cells. Does it mean there's lots of energy available? Does it mean there's not a lot of energy available? Does it mean the cells need energy? Seeing the responses coming in? Generally this is about right. You're seeing, as we mentioned, acetyl-CoA and NADH are going to inhibit it. We saw that through the feedback inhibition a couple slides ago. Pyruvate is going to indicate there's lots of pyruvate available. So PDC is going to be responsible for converting pyruvate into acetyl-CoA. So when there's lots of pyruvate, you'd expect that process to be turned on to use up that pyruvate. So lots of substrate around. ADP is going to indicate low levels of energy. So it's probably going to indicate that there needs to be a flow of glycolysis into the TCA cycle. So it's going to be turning on this process. So we want these enzymes turned on when there's low energy. Calcium is one we're going to see a lot as well, and this is more particular for muscle tissue. As calcium levels build up, that's part of a signaling cascade that's going to indicate muscle contraction or the need for work to occur. That's going to stimulate energy production. It's part of the reason why before you do any exercise, you do kind of a loose warm-up, get your muscles ready. That starts stimulating your metabolic processes to get fuel, your energy metabolism and your fuel systems working to allow you to exercise. So looking at this process, as we mentioned, acetyl-CoA and NADH inhibit the pyruvate dehydrogenase complex. Now the kinase, we talked about a second ago, this PDC kinase will turn pyruvate dehydrogenase from its active form to its inactive form. So let's write this inactivates, and this activates. So if we want the pyruvate dehydrogenase complex turned off, our kinase here is our friend. It's going to help us turn it off as well. So it's going to be stimulated. We want to turn this enzyme off to turn off the pyruvate dehydrogenase complex. Conversely, the phosphatase is what's going to activate this process. It's not what we want. So we want to turn this enzyme off. So you start to see how regulation of these substrates are going to have an impact on not just the enzyme itself, PDC, but also its regulatory enzymes. For pyruvate, pyruvate is going to indicate there's lots of substrate coming through. So it's going to turn this enzyme on, PDC on. And likewise, if we want to turn it on, we're going to inactivate the kinase, and we're going to potentially stimulate the phosphatase. ADP, that's going to indicate low energy states. We're going to want to turn on this process to get energy into our cells to generate more ATP. So you're going to see that inhibiting and activating as well. And likewise, calcium is going to indicate muscle contractions occurring. We need energy. So we're going to inhibit the kinase and activate the phosphatase. So let's look at those muscles at rest and see what's happening here. At rest, the NADH and acetyl-CoA levels are going to be very high. Cells are not using energy and not using these resources. So you're going to start seeing that kinase turned on to turn off this complex. So disconnect glycolysis from the TCA cycle. You see these molecules have the direct impact on PDC, binding to E1 and E2. And then this phosphorylation is going to have the impact on E1. So every enzyme within this complex is going to be inhibited, turning off the whole complex. And it's important to turn off E1 because that's the decarboxylation step. That's the one that's going to really be driving this reaction forward. So when you're exercising now, thinking about what this means in terms of the energy state of the cell and those molecules that are around. Exercising is usually going to indicate lots of glycolysis has started, lots of pyruvate is building up, but ADP levels are also going to be very high. Both of these are going to turn on the complex, and then we're going to see calcium as well having an impact on activating PDC as well through activating the phosphatase. These have the net reaction of activating the enzyme directly, but then also keeping it in its dephosphorylated or active state. So all these processes converge to ramp up production of acetyl-CoA from this enzyme. So we see all of these kind of working together. So depending on their levels, they may be a little bit high, they may be very high, depending on the needs of the tissues at any given time. And as the concentrations begin to increase, the activity will increase proportionally as well. So thinking about regulation and just summarizing some of those regulatory mechanisms we talked about. Product inhibition is a common theme we've seen with glycolysis. We're seeing it here as well. So acetyl-CoA and NADH binding to E1, E2, and E3 and inhibiting the enzyme. Covalent modifications, or PTMs, that are occurring on the enzymes. These are going to cause changes in the conformation, inactivating or activating the structure, depending on what that specific phosphorylation event is going to do. And then allosteric control as well. So this is going to be the products of the reaction, acetyl-CoA, NADH, pyruvate, and ADP, all are going to have an allosteric mechanism whereby binding to not only the active sites, but also to other allosteric sites in the enzyme, causing that change in that conformation, allowing it to work efficiently. And then calcium, again, stimulating that phosphatase, turning on this process, allowing energy to flow from glycolysis in the cytoplasm into the TCA cycle to ramp up that energy production in times of need. Now, acetyl-CoA is critical. Acetyl-CoA is useful because it's really at the center of all energy metabolism within the cell. Every metabolic fuel can be converted into acetyl-CoA. This makes it central in generating energy from any dietary source. Fatty acids can be converted to acetyl-CoA. Ketone bodies can be converted to acetyl-CoA back and forth. Carbohydrates get converted to pyruvate in glycolysis. Pyruvate gets converted to acetyl-CoA in the PDC. Amino acids can be converted into pyruvate. We saw alanine as being a source of energy, or being converted to pyruvate directly, and that can enter the TCA cycle through acetyl-CoA. And then ethanol. Ethanol is actually a very high energy molecule, just in general. As a side note, you get about 9 kilocalories per gram from fats. From carbohydrates, it's about 4 kilocalories per gram. Amino acids is about 4 kilocalories per gram. Ethanol, or an alcohol, is about 7 kilocalories per gram. You get a lot of energy from ethanol. So all of these funnel into acetyl-CoA. Now that acetyl-CoA, to use it for energy, it enters the TCA cycle. You might have seen this referred to as the Krebs cycle, the citric acid cycle. Here referring to the tricarboxylic acid cycle, the TCA cycle. All of these are interchangeable. Using your preference, you're not going to be docked for using one name or the other. Whichever one you're used to using, you can use within this course as well. But just know that we're going to be referring to it as a TCA cycle. Now this is where that oxidation of all these fuels occurs. That acetyl-CoA is going to undergo decarboxylation and oxidation, generating carbon dioxide. Remember, that's a driving force, pulling this reaction forward, and allowing and generating reducing equivalents, or NADH molecules, that will enter into the TCA cycle. So this TCA cycle is what completes that fuel oxidation, allowing those molecules now to go into that oxidative phosphorylation in the electron transport chain. Now, like with glycolysis, I'm going to give you a heads up on what I expect you to memorize in this course for the TCA cycle specifically. You need to know the structure of acetyl-CoA, the short form, not the long form that we showed. So just know the acetyl group plus the coenzyme A kind of short form. You need to know the structure of oxaloacetate and the structure of citrate. This is important because this is the first reaction within this process that gets the process going. You need to know the total substrates and products of this reaction. So what is the net substrates or input and what's the net output of this process, one round of the TCA cycle. And you need to know how it's regulated as well. So which enzymes, or where are the regulatory points, and what is going to regulate those. So looking at the TCA cycle, we can break it down into two different parts. The first part is going to be that oxidative decarboxylation, where we're going to be taking those two carbons that are coming from oxaloacetate and citrate and generating, releasing carbon dioxide to drive this reaction forward, generating NADH molecules. Looking at this pathway, oxaloacetate is a 4-carbon molecule, acetyl-CoA is a 2-carbon molecule, making that 6-carbon molecule of citrate, or citric acid. The reason we call this a tricarboxylic acid cycle is because citric acid has 3 carboxylic acids. Through this process, there's going to be some isomerization occurring, but there's going to be some key dehydrogenation reactions occurring at this first stage. This is going to release carbon dioxide, driving the reaction forward, and generating and storing that energy as NADH. That NADH is what's going to end up in the electron transport chain to generate energy. Counting the carbons, that's the 6 carbons that were initially in citrate and isocitrate, will end up being 5 carbons in alpha-ketoglutarate, and eventually 4 carbons in succinyl-CoA. So we've lost 2 carbons. We have a 4-carbon molecule now in a succinyl-CoA. Because this is a cycle, we now need to regenerate oxaloacetate. Sorry, and just as a note here on an example of what these dehydrogenation reactions occur, we call them a dehydrogenation reaction because we're taking a hydrogen with its electrons and transferring it to something else. So in this case, we're taking the hydrogen here from isocitrate, transferring it with its electrons to NADH, or NAD+, making NADH, and resulting in the decarboxylation occurring and generating alpha-ketoglutarate. So now that we have a 4-carbon molecule in succinyl-CoA, the second stage is all about regenerating oxaloacetate. This is a cycle. You need to come back to oxaloacetate at the start. So this is a series of reactions to help to reorganize the succinyl-CoA to generate this 4-carbon oxaloacetate. Through this process, we also generate some reducing equivalents, FADH, FADH2, and NADH, that are going to help fuel ATP generation in the electron transport chain. Key notes about some of the products of this reaction. A GTP is being made. Remember, a GTP is just a nucleotide triphosphate. It's energy. Like UTP, like ATP, CTP, TPP, these can be interchanged with one another depending on the needs of the cell. The cell will maintain a balance of all these nucleotide triphosphates in order to allow different metabolic processes to occur. So whenever you see a GTP, it has the same amount of energy as an ATP, and we can consider them essentially identical. There's also 2 water molecules used within this reaction. That first reaction with citrate synthase, and then later on with fumarase as well. So you need 2 water molecules to allow this process to occur. But overall, we're going to generate or transfer pairs of electrons to these reducing equivalents. In total, we're going to generate 3 NADH molecules and 1 FADH2 molecule. These electrons will move to the electron transport chain, where it's going to be another redox reaction, allowing us to fuel ATP generation. Succinate dehydrogenase is one enzyme I do want to call out specifically because it has a dual function. Whereas all these other enzymes are free within the mitochondrial matrix, succinate dehydrogenase actually has a dual purpose. It's a membrane-bound protein that's part of the electron transport chain. So not only is it part of the TCA cycle, it is directly involved in the electron transport chain. The first part of the reaction is that dehydrogenation reaction of succinate to fumarate. And the second part of the reaction, that FADH molecule, which is a prosthetic group, is bound to the enzyme, is going to transfer those electrons to coenzyme Q and enter the electron transport chain. So we're going to see this again on Thursday, but I do want to call this out right now as well. Overall, looking at this reaction, we can summarize the 8 steps of the reaction in this cycle and look at it from a thermodynamic lens. Under standard conditions, these reactions are all over the place. Some are energetically favorable, some are very unfavorable. But when we look at them within a cellular context, where these enzyme reactions are being coupled together, the product of one reaction becomes the substrate of the next reaction, it can pull this reaction forward in the forward direction. We see lots of energy being generated or released through some of these steps, so citrate synthase, isocitrate dehydrogenase, alpha-ketoglutarate, and even steps that under standard conditions are very unfavorable. When we link it together, an oxaloacetate now gets used in the next step of the reaction, and ADH gets used in the electron transport chain, we can make this unfavorable reaction essentially a reversible reaction under cellular conditions. So by coupling these reactions together in this cycle, it pulls it through and allows us to generate lots of NADH molecules in the process. Also note that those favorable reactions are the steps that are going to be regulated. And we're going to come back to this in a few slides. So think about which steps will be regulated here. So trying to summarize this, the TCA cycle, what is the net reaction for one round of the TCA cycle? So we'll come back to the last question, but now I want you to take a moment and try and answer these. So I'm going to give you about, say, 2 minutes. Look through that pathway chart and try and summarize, or look through even this chart, and try and summarize what the final reaction, the substrates and products of this reaction are going to be. I want you to think about where the carbon dioxides originate, and then jump into the Menti and look at those structures just from a cursory lens, and think about if any of them can be made from amino acids. So I'm going to give you a couple minutes to work through that. I'm going to walk around and help anyone who might need it as well. Oh. All right, turns out I forgot to set up the Menti for this one, so we'll just take it up together as a group in a couple minutes. All right, so coming back to this, is anyone still working through it? Need another minute? I'm happy to give you another minute. We'll go through it. So let's think about what is the main input? What is the first thing that's going to be reacting within this pathway that's entering into the pathway? It's coming from pyruvate dehydrogenase. It's going to be acetyl-CoA. So we know there's going to be acetyl-CoAs that come in. We know the products of the reaction are going to be NADHs. So in order for NADHs to be the product of the reaction, we need NADHs as substrates as well. Likewise for FADH2 is the product of the reaction. That means we're going to be a substrate of reaction as well. We have some water molecules we highlighted earlier. We have a GTP. So if we have a GTP that's coming in, we need GTP to start with. So we can start to think about where all these molecules are coming from. So if you were to put this all together, you would have seen 1 acetyl-CoA plus 3 NADHs, or NAD pluses, plus an FAD, plus a GDP and a phosphate that will eventually become GTP, plus those 2 water molecules will give a net output of a coenzyme A. So we need to break acetyl-CoA apart to generate those 2 carbon dioxides. And we have left over with that coenzyme A 3 NADHs, 3 FADH2s, 3 protons, and then we have our GTP as well. So where does the carbon dioxide originate? So looking at the TCA cycle, this figure has those carbons highlighted. Those 2 carbons here from, let me see if you, those 2 carbons here, from acetyl-CoA, if you were to track them through the process, they are not the ones that are going to be lost as carbon dioxide. The carbon dioxides that are lost through one round of the TCA cycle are actually coming from oxaloacetate. This means that on the next round of the TCA cycle, those carbons that are coming from acetyl-CoA, that's where they're likely to be lost in the second or third round of that TCA cycle. But in one round of the TCA cycle, those carbon dioxides are coming from oxaloacetate. The acetyl-CoA is helping to regenerate the oxaloacetate by replacing the 2 carbons that are going to be lost. And then which TCA intermediates can be made from an amino acid? Looking at the structure, there's 2 molecules that you should start to highlight. If you see a feature that jumps out at you, we saw this with pyruvate and alanine. Whenever you see a carbon dioxide or a carboxylic acid group next to a carbonyl group, that should be ringing or should be highlighting to you that this can be partially part of an amino transferase reaction or amino group. If you were to swap this with an amino group, which amino acid is that? Anyone want to shout it out? Hearing whispers, aspartate or aspartic acid. Likewise for alpha ketoglutarate, we see that same structure. What does that one kind of look like? We have our carboxylic group, we have our amino group, 2 carbons, and then an acid group. This should be glutamate, glutamic acid. So looking at this process, the TCA cycle is very useful in that it kind of becomes a hub for all of these different metabolites to enter and exit from the pathway. We call it an amphibolic pathway, meaning that it's both anabolic and catabolic. We're going to build this up and we're going to break these down. Oxaloacetate and alpha ketoglutarate are 2 main points where amino acid metabolism is going to feed into this pathway or kind of be extracted from this pathway. Other molecules within this pathway, malate, you're going to see it come up again in gluconeogenesis. Citrate, we're going to see in fatty acid synthesis as well as acetyl-CoA. And then succinyl-CoA is important for heme synthesis, so making that heme prosthetic group that you're going to see in hemoglobin and other enzymes as well. So this helps to pull things in and out of this pathway. And many pathways are going to be occurring. You're going to see metabolites from pathways being pulled out and coming back in, depending from different sources as well, to potentially be used as fuel or to potentially be used as building blocks to make other molecules. Now, I have a question for you, and this one I did set up the Menti. Which steps do you think are going to be regulated within the TCA cycle? Which steps are going to be regulated in that TCA cycle? Or would you predict are going to be the regulatory steps? So seeing the steps coming in, steps 1, 3, 4, the irreversible ones, anything that releases CO2, 1 and 4, yes, don't forget 3. All those irreversible steps. So anything where you see that high negative delta G should be indicating that the cell is going to find a way to regulate the step. This is an irreversible step and needs to be turned off. It's not going to be controlled by substrate concentrations. So looking at this regulatory, how this process is going to be regulated, we see that citrate is going to be product inhibition of citrate synthase. As citrate levels increase, it's going to turn off that pathway, allowing citrate to be used for something else, and acetyl-CoA to be used for something else as well. Thinking about it in general, anything there's low energy, or when there's a high need for energy, so calcium, is going to indicate it's going to turn on this pathway. So ADP is going to indicate low energy, calcium is going to indicate low energy, and it's going to turn on this pathway to allow the cell to generate more energy as needed. However, if there's lots of energy available, this pathway is going to get turned off. And that's where you're going to start to see NADH. NADH is going to be indicative of energy in the cell. This ratio of NADH to NAD plus is a surrogate for the energy ratio within the cell. Lots of NADH, there's lots of potential ATP available. We'll turn off this pathway. There's lots of NAD plus, we need to turn this pathway on in order to generate more energy to sustain homeostasis in cellular processes. Now, malate dehydrogenase is also regulated by NADH. This is product regulation, so the product of this pathway will turn it off. And this is an important step because it allows oxaloacetate to potentially be, or malate to potentially be used for another process. And you're going to see that again a little bit later when we talk about gluconeogenesis. I've been foreshadowing to a lot of conversations you're going to have with Dr. Patterson over the next couple of weeks, because all these pathways are interconnected. We can't look at them in isolation. They are all going to feed into one another and regulate one another because they're so integrated into generating energy for the cell. So think about what it means when the cell has lots of energy and what it means when the cell needs energy and which pathways are going to be turned on and turned off. So in summary, we see the TCA cycle as this hub of energy regulation. It's where this oxidation of acetyl-CoA is going to occur, connecting the breakdown of all these metabolic fuels. Overall, the pathway is going to be very favorable. Despite that really positive delta G for the malate dehydrogenase, it's still going to pull it through because oxaloacetate, acetyl-CoA, the use of NADH is going to pull this pathway forward when energy is needed. That regulation is going to occur at all those favorable reactions. And this pathway is going to be what we call amphibolic, meaning that it's going to be used to generate energy, but also used to create new things because many of the molecules become metabolites as well. Now I have a question for you just before we wrap up. We've talked a lot about the vitamins or those cofactors that are present within the enzymes. If anyone here takes a supplement, some sort of a vitamin supplement, a multivitamin, or anything else, where you see any dietary supplements, you might see claims like B vitamins experience a natural boost in mental and physical energy throughout the day with their slow-release capsules, feel refreshed and rejuvenated as you go about your daily activities. Maybe you might be thinking, hey, that Mentimeter when we checked in, I saw a lot of people were saying they were pretty low energy. Maybe I'm just going to take a supplement and it'll help me out and give me an energy boost. How many people think this is realistic? Oh. How many people think that this is accurate? A lot of people that, I don't know about this pile. But there's some people who are thinking yes. A lot of people think, good chunk thinking no. A lot of people somewhere in between. And in general, it's probably closer to something in between. So looking at these vitamins, these cofactors, they are all going to be cofactors for these metabolic processes. The B vitamins we often talk about are B1 to B5. So thiamine, riboflavin, niacin, biotin, pantothenic acid. And they're going to form ATP, FADH, NAD, biotin, and coenzyme A. If you don't have enough of them, many of these enzymes will not function. So if you're deficient in these enzymes, these processes of energy metabolism are going to stop. And we see these with the effects of deficiencies, enzymatic or nutritional deficiencies in these enzymes or in these cofactors. They result in a lack of energy, poor wound healing, dermatitis, dementia, nausea, lethargy, depression. All of these can be traced to decreased energy output, but only when you have a dietary deficiency. If you are getting enough of these enzymes in your diet, it doesn't matter how many of these you take, you are not going to get more energy. We get energy out of those fuels, carbohydrates, fats, amino acids that you consume in your diet. The vitamins on their own cannot provide energy. They can allow you to extract energy, but if you have enough of them, they're not going to give you any boost beyond normal. So they're not going to improve anything. They're just going to bring you up to baseline if you're deficient. So key messages for this week, think about how we integrate these pathways together and what happens to these fuels when we don't need them. What happens to glucose, and you're going to see what happens to fats and acetyl-CoA next week. Think about how these processes are connected, what's connecting glycolysis to the TCA cycle, that's the PDC, and how we regulate these pathways and turn them on and turn them off together. We'll see you next week in a couple minutes. It's 11 o'clock on the 11th day of the 11th month, so just take a moment and think about what Remembrance Day means as well. So thank you. Take care.